Hello, my name is Valeria. I would like to introduce a study that looks at epigenetic mechanisms relevant to Parkinson's disease. Parkinson's disease is the second most common neurodegenerative disease in individuals over 65 years old. It manifests as dopaminergic neuron loss in substantia nigra pars compacta, situated in the midbrain. And these neurons have a distinct dark appearance, as seen on this cross-section of the midbrain. The proposed mechanism for dopaminergic neuronal death is through upregulation of protein kinase C delta. In previous studies, it has been shown that PKC delta inhibition attenuates neuron toxin-induced cell death, while PKC delta overexpression promotes loss of dopamine production and leads to neuronal apoptosis. Although post-translational regulatory mechanisms of PKC delta have been documented, epigenetic changes altering PKC delta expression have not been investigated. To investigate whether PKC delta is regulated epigenetically, various assays modifying histone acetylation were used. It appears that hyperacetylation of the genome upregulates PKC delta expression, and this increase in PKC delta expression promotes pro-apoptotic pathways in dopaminergic neurons. To look at PKC delta expression in relation to histone hyperacetylation, cultured primary mouse neurons were treated with different histone deacetylase inhibitors, and PKC delta expression was quantified by Western blotting. To validate whether histone acetylation is directly responsible for PKC delta upregulation, histone deacetylase inhibitor-treated neurons and untreated neurons were used in chromatin immunoprecipitation assays. Acetylation of histones at the PKC delta promoter region were investigated. Finally, 6-hydroxydopamine was used to induce oxidative stress in neurons, and their viability was quantified. Untreated neurons, hyperacetylated genome neurons, and hyperacetylated genome neurons treated with PKC delta inhibitor, along with adequate controls, were used. Hyperacetylation of the genome through inhibition of histone deacetylase upregulates protein kinase C delta expression. On these Western blot gels, you can see the PKC delta expression in neurons. There is a time and concentration dependent increase in PKC delta when neurons are treated with various different histone deacetylase inhibitors. The result is the same. Increase in acetylation is followed by an increase in PKC delta. Histone deacetylase inhibitor, sodium butyrate, increases acetylation of histone 4 at the PKC delta promoter region. Nuclear material collected from sodium butyrate treated neurons contained hyperacetylated histone 3 and histone 4. Analysis of DNA that was associated with the hyperacetylated histones revealed the presence of the PKC delta promoter. In dopaminergic neurons, overexpression of PKC delta by hyperacetylation sensitizes the cell to apoptosis by oxidative stress. Exposure of neurons to oxidative stress by 6-hydroxydopamine reduces their viability. This effect is enhanced in hyperacetylated neurons. However, treatment with PKC delta inhibitor rotlerin attenuates the neurotoxin-induced apoptosis. It was shown that PKC delta expression can be regulated epigenetically in dopaminergic neurons via histone acetylation in a manner that is consistent with the known mechanisms of cell death in Parkinson's disease. For it remains to be investigated are the direct interactors of PKC delta and the detailed mechanisms of their pro-apoptotic signaling. Mapping cellular events and their regulatory points in relation to cell death is monumental in understanding and treating neurodegenerative diseases. Here, the authors present several potential therapeutic targets that will reduce the neurotoxic effects of oxidative stress in neurons, as seen in Parkinson's disease. Thank you for listening. I hope you found this really interesting.